All right, good evening, everyone. We'll go ahead and get started. Welcome to our third Life Science Lecture Series for our 20th anniversary celebration. My name is Sarah Brooker, and I'm the Program Coordinator here at Colorado Canyons Association. If you're not familiar with CCA, we're a local nonprofit located here in Grand Junction, and our mission is to foster community education, stewardship, and awareness of our 3 national conservation areas in western Colorado, those being McInnis Canyons, Dominguez Escalante, and Gunnison Gorge, NCA. So basically what all that means, we love getting people outside and connecting them to their public lands. This year marks a pretty special year for McInnis Canyons. It's its 20th anniversary. So on October 24th, 2000, Congress officially designated McInnis Canyons as a national conservation area, which permanently protected the landscape. I'm sure a lot of you have enjoyed the resources out in McInnis and have done a lot of hikes and boating trips out there. And so we want to celebrate the 20th year with you all by offering some community hikes, rides, and lectures throughout September and October. So that brings us to our third Life Science Lecture Series. Tonight we're here with Dr. Aparna Palmer. She's a professor of biology at Colorado Mesa University and has done a lot of research out on our public lands, particularly in regards to water bears. So I know I'm really excited to learn more about these creatures. If you have any questions during the lecture, you can type them in the questions and answers box, and we'll leave about 10 minutes at the end to go through those questions. So without further ado, I will hand it off to Dr. Palmer. Thank you so much for being here this evening.
Thanks. Thank you, Sarah. It's a real honor to be here, and thank you everyone for letting me share one of my passions, which is talking about and working with water bears. As Sarah mentioned, I am a professor at CMU. I'm also currently one of the assistant vice presidents for academic affairs. So in that position, it takes me a little bit further away from the research lab, but I still find time to hike the canyons, which is one of my favorite things to do. And I was there just yesterday, actually. And I also love being in the lab. So I hope that you enjoy what I share with you. I'm going to go ahead and share my screen with you. It'll take me a few moments to load my presentation, and then we'll get started. All right, well, today I'm going to talk to you about the water bears of the Colorado Canyons. And as you can see by my exclamation point, I find them to be microscopic and yet magnificent. So these are some pictures of some water bears that occur all over the world. They are beautiful, beautiful organisms. And then a couple of pictures that my students and I have actually taken in the lab. Today I'm going to talk a little bit about the work that we're doing in the Devil's Canyon area. And so I want to make sure and give credit to the student that I worked with a few years ago. She is now an alumna of CMU, Miss Beth McBride. She actually worked for the BLM while she was working with me, and she hiked the Devil's Canyon area with me and also without me to collect these beautiful organisms. So I want to point out that these organisms come in a kind of a wide array of different sort of shapes and forms. One of the craziest forms is shown on the left side of your screen. As you can see, sprouting out of this beautiful little critter are some leaf-like structures. Those are actually gills, and that one is found in the ocean. The rest of them are actually found inland, but they are all aquatic. So I'm going to talk a little bit about them for those of you who... So water bears fall into a special group of organisms called the tardigrades. And the entire group of tardigrades falls into a phylum, which is a category within the animal kingdom. And that phylum is officially known as phylum Tardigrada. As you can see, it was established in 1767, which is close to the invention of the microscope. So the microscope enabled us to see some organisms and discover animals that we had never seen before. These organisms were called water bears or moss piglets. They are organisms that require water in order to exist. They are all microscopic, and the largest basically gets to about 1.5 to 2 millimeters. And so on average, they're about 1 millimeter in length, which means that if you could see them with the naked eye, they would look like the dot that you would make with a pencil. They are found in a wide variety of habitats. So they're found in the world's oceans. They are found in ponds and lakes and rivers. And interestingly, they're found in what are known as semi-terrestrial environments. And those are the environments that we found out here in the deserts. One of the things that's most interesting about them is that many of them are found in what are known as extreme environments. So they actually can live and they can thrive in extreme environments, and so we can regard them as what are known as extremophiles. So here on the left, we see a picture of one that was captured by one of my students. This is a water bear that occurs around here, and it actually represents a new species that we are currently describing. So before I dive in and talk about water bears in more depth, I want to talk a little bit about how unusual it is to find water bears here in our environment. So obviously we live in the high desert environment. I've taken a couple of pictures of the Devil's Canyon area to remind you of how beautiful it is, but also to remind you that it is a high elevation desert. Clearly, we have a lack of moisture with very little rainfall. We have very, very high light intensity, and in fact have quite a bit of UV radiation. The rainfall that we do get is unpredictable and quite variable. And as you know from living out here, the temperature extremes can be quite dramatic. And so you can start out the day with freezing temperatures. In the afternoon, it could be a comfortable 70 degrees. And then you could go into nightfall and it could be very, very cold again. And so if you're going to survive out here, you really have to be able to be adapted to those extremes. The landscape is quite striking and beautiful. I've lived out here for about 21 years now, and every single time I go out there, I think, gosh, I live in one of the most beautiful areas in the entire world. So every day we get to see red rock canyons, we get to see mesas, we get to see these beautiful washes that you can walk up. And of course, the vegetation is spectacular. Things like these old junipers, pinyon pines, rabbit brush, Mormon tea, and of course, the ever-present sagebrush. Now, you might wonder, because tardigrades are aquatic, where in the world would you find these organisms? Because if you take a walk out there, for example, I took a walk out in the Devil's Canyon area yesterday, you might wonder, where's the water? And so around here, of course, you can look for water in the river, and occasionally when it rains, you can look for what are known as ephemeral waters, like a little pothole. But actually, a lot of the water that we find after it rains is trapped in what is known as biological soil crust, and sometimes a component of that crust, and sometimes existing alone, these organisms called mosses. And so here I have a picture of the crust occurring on the edge of a rock, but I also have a picture of mosses occurring alone on a rock. And I'm going to focus on mosses, but I want to take a moment to talk about both crust and mosses and what they offer. So crusts and mosses are interesting because they actually host a wide variety of organisms. In fact, they host an entire community of organisms. And they're able to do so because of the environment in what is really a very harsh environment, the desert. And they're able to do that because they anchor soil particles to one another. When it rains, they actually absorb and retain a huge amount of water. And so if you have an area of soil crust or even just mosses, The runoff of water is much lower than it would be in other areas, and so they reduce erosion. Soil crusts in particular have organisms in them that can actually fix nitrogen and make the soil richer, excuse me. They also aid in decomposition because they have organisms that actually break down organic matter. Organisms that live in there have protection from the UV radiation that occurs out here. And they also, on those really cold days, can absorb heat and be a very comfortable environment for that community of organisms that lives within the crust and also lives within the mosses. So what kinds of organisms are associated with these crusts and mosses? Well, first of all, the organisms that we are able to detect include, of course, bacteria, but they also include fungus, they include algae, they include liverworts, and they also include lichens, which are an association of fungi and algae. And so in this picture, you can actually see some mosses, but then associated with the mosses, you can see some lichens, which are bright and colorful, that occur out here as well. But there's more than just that. And it really has to do with the fact that it occasionally rains. So while this habitat is susceptible to high rates of evaporation and extremes in daily temperature and changes in salinity, it is actually a temporary aquatic habitat every time it rains. And so I'm one of those people, knowing that this is a temporary aquatic habitat, I just get really excited when it rains. And I always rush over to the window and I tell anyone who's around, including my poor husband, that when it rains and the mosses are able to soak up all of that water, that the tardigrades are waking up. And so is an entire community of organisms that is living within those mosses. And so I'm going to explain what that means in a couple of minutes. So the fauna of these habitats, particularly the mosses in this case, which is what I study, is really rich. So if you were able to look at these really lush green mosses under the microscope, you would find kind of an entire ecosystem. And these ecosystems include not only like unicellular organisms, it also includes multicellular organisms, things like flatworms and roundworms. I know flatworms and roundworms get a bad rap because most of us think of them as parasitic organisms, but there's some good ones too. They're out there called free-living flatworms and roundworms. We also get some cool critters called rotifers, which are seen right here and a little bit right here. Rotifers have this really cool wheel-like apparatus that they use to collect food. Gastrotrichs, here's a gastrotrich right here. These are both multicellular animals that live within the mosses, and they're both microscopic. We have mites and spiders and insects, millipedes and centipedes, but of course, included within are the creature that I focus my study on, the tardigrades. And here's an example of a tardigrade right here. They look like these tiny, beautiful bears that are creeping and crawling among the forest that is the moss within the desert. So water bears are like other animals. They have an external covering. The external covering is called a cuticle. The cuticle is somewhat hard. It's not real hard. In fact, air can actually go through the cuticle, but water can't. They have 4 pairs of leglets. So they're little legs, so we call them leglets. They have specialized feet that include these little claws or sometimes little suckers. They move using muscle. Now, our muscles work against our skeleton, which is bony. Their muscles work against fluid in their body, and so we call it a hydrostatic skeleton. They also breathe. They actually breathe directly through their cuticles, so air is actually entering through the surface of their body and then exiting that way as well. So oxygen and carbon dioxide are exchanged directly through their skin, which is, you know, these are some of the processes that all animals take part in. I wanted you to kind of see what a water bear looks like on the inside, and I wanted to point out some of the things that the water bear has that I'll talk about a little bit later. So if you were able to kind of look close up at a water bear, you would see that it's an animal with a mouth, and yes, it has an anus, so that's similar to us. It doesn't have eyes, but it does have an eye spot. And an eye spot then allows it to tell the difference between light and dark, even though it cannot form an image. It has that external covering called the cuticle. Notice it has these little claws. It has an entire digestive tract. So from the mouth, the food would go into this little tube called the buccal tube, kind of like a little throat area. From there, it goes into what's called the pharynx, which is a muscular area where the food is processed. It has a gut area as well. Notice that this particular specimen has some eggs that it's going to produce, so it's a female. And so it has a number of different structures associated with that. And it has a complete gut, which means it has both a mouth and an anus. And so I'm going to come back to some of these structures a little bit later, but I wanted to kind of show you that it has a lot of things that most animals do. So now I want to show you a quick film of what this guy looks like under a microscope. And so I want you to notice that it has these little eye spots, which are these dark areas. These areas right here and here are actually its salivary glands. This is its pharynx, which is a muscular structure that it uses to grind food. This is its buccal tube where food is going to enter. And then its mouth region is right here. Right here and here are these little stylets. Stylets are spear-like structures that it's going to use to poke whatever it wants to feed on, and then it's going to suck out the contents. And so just to give you a context to here, it's going to poke an individual cell and suck out the contents of the individual cell. That's how it makes its living. These organisms can be herbivores, but some of them can be carnivores as well. So I want to give you kind of a sense of what it looks like under a microscope. So what you're seeing kind of moving around within its body are particles that actually store energy. They're called food bodies, but actually they basically store an oil that the animal uses for excess energy. You can also see its stylets moving. So I want to talk a little bit about tardigrades and extremes because this is something that happens out here. So tardigrades are capable of a special process called anhydrobiosis. This is especially critical out here. So anhydrobiosis is a suspension of metabolism, technically called cryptobiosis, that's initiated when the environment starts drying out. So I want you to imagine a moss that is hit by rain. And so initially that moss becomes this really spongy green structure. It's an aquatic habitat. The tardigrade is able to sort of move around in there, enjoy itself, make its living. But eventually, because we live in the desert, it's going to start drying up. And as the moss starts drying up, the tardigrade, in order to survive, is actually going to start drying up as well. And so what's going to happen is it's going to go from this active form into what is known as a ton, which is its inactive form. This is the way in which tardigrades have survived out here for millennia. This is a special process that is amazing. So once they go into this special dormant phase, when they become a ton, they can withstand an environment that is basically devoid of water. They can withstand lack of oxygen. They can withstand really, really salty conditions, which kill most creatures. and notice the range of temperatures that they can actually withstand. Now, scientists have actually taken tons and subjected them to all of these extremes to actually test what they can withstand. One of the most fascinating things that scientists have done is they've taken tons and shot them into space just to see, for example, can they survive the vacuum of space? Can they survive anoxic conditions? Can they survive really, really high pressures? Things like that. So they've done all sorts of experiments, and they've found that within the ton phase, they can actually survive all of these conditions. Now, the creatures out here, really what they're basically withstanding is the arid environment, highly salty conditions, and of course the extremes in temperature. When it rains again, they can reanimate. So within a few hours of being exposed to favorable conditions, they can actually reanimate without DNA damage. Now, I put a question mark there because we need to do a little bit more research on that. But what's really amazing is that much of our research has shown that under, just normal conditions, if they can go into the ton phase slow enough, and if they can reanimate slow enough, they actually do not sustain any damage to their chromosomes. And so during their lifetimes, they can go into that dormant phase, come out, go back, and they can go back and forth several times during their lives, which is why they can survive out here in the desert, which is why I can go and collect a moss that's completely dry, bring it back into the lab, slowly hydrate it, and then have these water bears actually reanimate and crawl out of these completely dry mosses when I first collected them. So now I'm going to switch over to the project itself, and I'm really in the early stages of this particular project, and so I'm going to talk about where we have gone with this project and what there is to do, and then I'm going to talk about a similar project that I've conducted in the Colorado National Monument, because it's really important to compare and contrast this project with the other projects that I've been conducting on these same animals in adjacent areas. So the first goal of the project is just to look at which species actually exist in the Devil's Canyon area, and also establish new records, or maybe even establish new species for the Devil's Canyon area. Now, once we do that, it's really interesting to compare those data with adjacent areas. This is important. So, when tardigrades were first being studied, it was thought that they had a worldwide distribution. So it was thought that, if you ran into a particular species in the Himalayas of India, and then you ran into something that looked similar in the middle of Colorado, that they were the same species. It was thought that in this ton phase, they could actually fly all over the world, so to speak. Now we are discovering that their ability to kind of disperse themselves is far more limited than we ever thought. So limited that in an individual canyon, in a crack in the ground, it could be that in a small area, we might have one species, and then as you kind of hike maybe even a mile away or 2 miles away around a corner, you might find a completely different species. So the scope or the scale of speciation for these creatures is much smaller than we ever thought. And so when I first started working on these creatures, I thought, well, if I collect over here, and then I collect 5 or 10 miles away, it must be the same species. What I'm learning is they are absolutely not. They may look very similar to one another, but when you do the DNA work, you find out that they are not. And so there's a lot to uncover about not only who these species are, but how they may have evolved in an area where they're actually pretty close to one another. Okay, so now I kind of want to jump in and talk to you a little bit about how you actually study these organisms. So my student Beth and I, we actually started this study in the winter, went out to the Devil's Canyon area, these trails, we were looking for mosses. And now mosses are found in a couple of different ways. So you can look on bare rock for mosses, but you can also look in soil crusts. We did not want to disturb soil crusts because they are such delicate and important areas. And so we just decided to limit our study to mosses that were growing basically on bare rock. And so rather than just kind of looking in these areas that are adjacent to trails, we wanted to look at trail adjacent areas that were more accessible to bare rock areas. And so we kind of looked for these really beautiful mosses that were occurring right on bare rock here, or perhaps here, or you can see these mosses here. And so what we would do is we would just collect these mosses, we would record where we collected them, and we were doing a preliminary study. So just a preliminary sampling. We just wanted to see, okay, where are we going to find these tardigrades, and then go back and do a more extensive sampling once we kind of had an idea of what we were looking at. So I got to tell you that when you first hike these trails, you don't notice mosses. But now I guarantee you, when you go back and hike these trails, you will see mosses everywhere. And so it's kind of all of a sudden you sort of develop a search image for them, and then you see them everywhere. And so what I want you to remember is that they are everywhere, and living within these mosses are thousands and thousands of beautiful animals, including tardigrades. So once you collect those mosses, they have to be hydrated. So we bring them into the lab, and we have a special mix of water. The mix of water is spring water mixed with distilled water. Essentially what we're trying to do is replicate rain. And so we just soak those mosses for a couple of hours, about 2 to 5 hours in this water mix. And then we squeeze those mosses onto a sieve. The sieve is a very, very fine sieve. I mean, it's going to catch creatures that are as small as these creatures are. And remember, these are creatures that rarely get over 1 millimeter in length. And so on average, they're 0.5 millimeter in length. And so they're going to catch these types of creatures. And then once we filter them on that sieve, we wash the contents of the sieve into a petri dish, and then we hunt them and extract them using a microscope. And that's the fun part. And I remember, I've had lots of students go through this, and my best student actually was one who played a lot of video games. And I remember asking him, I was like, how are you so good at spotting these water bears? And he's like, Aparna, I think it's because I play a lot of video games. And I thought, wow, I wouldn't have guessed that. But once you develop a search image for them, as you're kind of looking through the petri dish, you can see them. And so they have a distinctive movement under the petri dish. They're kind of crawling real slowly. They're a little confused. They're like, where's the moss? I don't have anything to cling to. They're very, very cute. These creatures, they are not known to be parasites. They are all free-living organisms. As I mentioned, they're either herbivores or they are, a few of them are carnivores, and so they're not dangerous to work on in any way. So once we have them, we visualize them with light microscopy or electron microscopy. We collect lots of different types of data on them. We identify them using special keys. Keys, for those of you who are unfamiliar with them, are kind of like choose your own adventure sorts of, I don't know, pathways where you basically are given choices. Does it have this feature versus this feature? And based on your choice, you get to go down a pathway where it gives you another choice and eventually get to an identification, or you don't, in which case you might have a new species. And then you basically count the number of different species and you try to figure out where they exist and things like that. So I put a picture of one of my favorite water bears that's found out here. This is a water bear that is in the species complex because it's always bright red under the microscope. This one actually has these little dark structures in there, and those are actually its eggs. So it's actually ready to release some eggs to the environment. It's just an absolutely beautiful creature to see under the microscope. Now, here's what's interesting. So it's in its own species. So Echeniscus is the genus, Testudo is the species, but within the species, there are actually different species. And so the classification of these organisms is very, very complicated. So I wanted to give you a sense of what kinds of data you collect on these little bears. So when you collect data, you collect it under a microscope. So you have to make 16 different body measurements on each specimen. And I've given you some of these body measurements. For example, you have to make all of these claw measurements. You have to measure the buccal tube. And 10 of the body measurements are actually adjusted for size using the buccal tube length. And so it's actually a very painstaking procedure. And so you have to do hundreds and hundreds and hundreds of measurements. And then you have to apply some statistical procedures in order to really make them reliable and uniform. And just get the data that you need in order to make a determination about the species that you are looking at. So in addition to this type of data, which is called morphometrics, which then you have to apply statistics to, you also have to look at morphology. So morphology is just body shape and form. And so I've given you an example of Echeniscus testudo. And on these critters, you have to look at things like these like hair-like structures. You have to count how many there are. You have to look at these little horn-like structures. You also have to look at the plates that they have. So notice that they have these cool little armoring on their back. And so the shape of that armoring, how much they have, you know, where does it end, where does it begin? You have to look at all of that in order to help make the species determination. So with that, I want to give you just a quick overview of what kinds of species we find in the canyons. And so these are species that we find all over, not only in the Devil's Canyon area, but also in the Colorado National Monument and in some adjacent areas as well. And so I'm very proud of these pictures. These were taken on a special microscope at CMU. So we have, of course, my personal favorite, Echeniscus testudo, which is a species group or a species complex. We also have this critter, which is called Macrobiotis harmsworthii, another species complex. Macrobiotis hooflandii is another species complex. And then a rare one called Rhamazodius occurs out here as well. Now here's what's interesting. Even though I've given you the species epithet, Testudo, harmsworthii, hooflandii, they all represent pretty much new species. And that's because no one other than us has ever looked at tardigrades in Mesa County. And so other than a single student who worked on looking at tardigrades about 30 years ago, who looked at them but never really got a chance to identify them, this is really the first time that we've really tried to kind of figure out which species exist out here. And it's a very, very complicated case. And so to figure out who it is that we have, we're going to have to look at them very carefully and do DNA work. The work that we've done the most on is this guy. This guy is in the genus Milnasium. Now, Milnasium is an interesting critter. When you see Milnasium under the microscope, it's totally different from other tardigrades. So the term tardigrada literally means slow walker. This is the exception. This guy is the fastest walker of them. And so this guy actually moves really, really rapidly in the Petri dish. And what's interesting about this guy is while the others are all herbivores, this guy is a carnivore. It actually feeds on other water bears. So it's a very, very interesting creature. I want to give you a sense of the composition of the species that we found in the Colorado National Monument, which of course is adjacent to the Devil's Canyon area. We haven't completed our analysis of Devil's Canyon, and so we're interested in seeing if it'll have a similar composition. So in the monument, what we see most of is this guy, McInnis testudo, and we also see a lot of this carnivorous guy, Milnasium. And so I'm curious to see if we will find a lot of these two kind of species or new species within these two groups in the Devil's Canyon area. So far, we have found Echinuscus and Milnasium in the Devil's Canyon area, and then some Macrobiotis, but we need to do more sampling to really see what's most common in that area. So you can see this is 576 organisms that were collected to come up with this, which is a lot of work under a microscope. A quick note about genus Milnasium, the little carnivore guy that we've done the most work on. This genus has 35 species. We will be adding a few more species to this particular genus that come from this area. This is an interesting group because they are pseudocryptic or cryptic. And what that means is that they may be of two different species, but they look very similar. And sometimes it takes DNA in order to tell the difference. They have these really unique and interesting claws. They have primary claws and secondary claws. They are semi-terrestrial, as all of the water bears that we have been working on are, which means that they live in these mosses and they only come out when it rains. And as I mentioned, it is carnivorous. So this guy is about 0.5 millimeter in length, and what's interesting, what makes it different from the most similar species, it's essentially its twin, is that its fourth leg has really long claws. So this is the fourth leg, and here are its claws. The species in the world that it's most similar to is one that is found in the Himalayas of India. And 30 years ago, we would have said that they were the same species, but now we know they are not. They're very different, and it took a while, but we found that they were distinctive. And so some of the distinctiveness comes from the fact that these guys have a cylindrical buccal tube. They have a smooth cuticle. They have primary claws on all of their legs, as you can see from these electron microscope pictures. They have no accessory points, which are these hair-like structures on their primary claws. They have no bars at the bases of their claws, which are things that you tend to look at with these creatures, which kind of gives you a sense of how minute the details are that you have to look at. And as I mentioned, the species that it's most similar to is a species that was originally described in the Himalayas of India, Milnasium longiungue. And Milnasium longiungue has these accessory points, but our Milnasium, the one that we're studying right now, does not. And so we're eager to finish up the DNA work on this and publish this work. So further work, we know that there are several species of tardigrades in the Colorado National Monument. We know that some of these genera are represented in the Devil's Canyon area, but we need to do more work to kind of figure out how many total species are there in the Devil's Canyon area, and are there some new species in that area as well. And it's most likely that there are new species in addition to some new records as well, which means it is a species that maybe occurs in other areas of the West, but also occurs here, and it's the first record of it being here. So I want to finish up by just kind of talking about why I study tardigrades, other than just being a person who just loves life and loves small organisms, organisms that basically most people don't pay attention to. I really love the idea of contributing to our knowledge of species diversity on our planet. Our planet is filled with so many amazing species. The species that we know least about are the microscopic species. That's where the horizon is for biology. If you want to discover new species, it's all about going microscopic or going to the bottom of the ocean. And there are some tardigrades at the bottom of the ocean, by the way. This research can also help us understand extremophiles. And so if we can understand how an organism can adapt to extremes, it can actually help us survive as humans. So I want you to think about being an organism that can adapt to an extreme where you can basically become this thing called the ton. The ton is absolutely amazing. The organism loses 99% of its water in order to become a ton. It rolls itself into a ball. If you look at it from the cellular level, this organism actually packages itself in such a way that it can survive for 50 years in that particular package and then come out in a way where its DNA is not destroyed. So that means that that packaging could really teach us something about how to preserve tissues, or it could teach us about how to lengthen our lives. So, maybe there's a way that they're protecting their DNA as they go in and out of these dormancies that will help us survive better as humans. Maybe it will help us as we think about space exploration. So I think there's a lot that we can learn from tardigrades, and other than the fact that they are absolutely fascinating and really fun to look at under the microscope. I want to end by thanking a number of people and organizations. There's a lot of people and organizations to thank. I do want to point out again that Beth McBride was a key person who worked with me in the lab. She also worked for the BLM while she was here. She was very, very lucky, I think. And I know that there are other students who are able to work there and intern for the BLM here in this area. And for that, we at CMU are incredibly fortunate. It definitely was a key to her employment now. So I'm very thankful for that. So that's it for me. Thank you so much for your attention. And I'm happy to take some questions.
All right, thank you so much. That was so fascinating, and I know I learned a ton. I'm really excited to go out in McInnis next time and look a little bit closer at the moss and imagine all the tardigrades roaming around in there. So we do have quite a few questions that came in. I'll go ahead and get started from the top. You touched on this a little bit, but are tardigrades, are they in ton more often in these environments versus coastal environments?
Oh, that's such a good question. I love that. So in general, yes, tardigrades that live in semi-terrestrial environments such as mosses definitely have these adaptations more so than marine tardigrades, with one exception. So those tardigrades that live like in a tide pool, so right where the land meets the ocean, that's actually considered an extreme environment because it's impacted by the tide. So as the tide goes in and out, it's actually subjected to high rates of desiccation, high rates of UV, like terrible temperature fluctuations. And so the tardigrades that live in that environment actually have these adaptations, but they can go into the dormant phase in response to high salinity and freezing and stuff like that. So they have some cool adaptations. But in general, if they live in a stable marine environment, they don't have these adaptations.
Okay. So we have a couple folks who are asking, what is the average life expectancy of tardigrades?
Oh, that's a great question. So, you know, if the organism did not go in and out of the cryptobiosis, the dormancy state, it's probably just a few weeks. However, because they do go in and out, you know, no one has completely measured it, but it's probably a few years to a few decades. The kind of longest lived tardigrades were reconstituted about after 100 years of being in a ton state, but they didn't survive very long. And so, you know, as long as they're carefully preserved, they can survive for decades.
Great. All right, we have a couple questions from Annie. The first one being, do you think similar habitats in Delta and Montrose counties have tardigrades too?
Yes, they definitely do. I've had students study tardigrades in Delta County, and they have found the Echinuscus and the Milnasium. And so, oh, Beth, is Beth here? Anyway, yes, they definitely, they're all over, you know, the western slope of Colorado, and definitely, you know, Delta and Montrose counties have similar habitats, and so they would have that.
All right, and her second question is there a limit on how many times they can go into the ton phase and reanimate?
Nobody really knows the answer to that question. So, you know, the ton phase, going in and out of the ton phase, that is a delicate process. So they can't go into it very quickly. You know, it has to be somewhat of a slow process. So it has to occur over a few days. They've tried to replicate it in the lab, and if you dry them out too quickly, they aren't successful in going into the ton phase. So we don't know the answer to that question.
Okay. Next question. Do the young tardigrades that come out of eggs look like small adults, or is there a larval form?
So, you know, they can have a kind of a larval form. I think what we would call them are juveniles because they closely resemble the adults. However, they can have fewer limbs or differently shaped claws. And there are different patterns when you look at marine tardigrades versus the semi-terrestrial or the freshwater tardigrades. But they look like miniature versions of the adult, except that in some species, they can have fewer limbs or different claws.
All right, question from Derek, and he's asking, are tardigrades so diverse because they've survived so many mass extinctions, or would they have evolved regardless?
So it's hard to describe the effect of a mass extinction event on tardigrades. It really depends on the pace of that event. The diversity of tardigrades, you know, in terms of the total number of species, I have to tell you there aren't very many tardigrade researchers in the world. So we probably have an underestimation of the number of species. The last time I was at a world tardigrade conference, there were 90 of us. So there's a lot of work to do. Right now, you know, we're around 1,500 to 2,000 species, but it's probably 10 to 15 times the number of species. In terms of the speciation events, their diversity, it's probably due to kind of localized adaptation. And really we really have to try to understand like how micro environments can push change. And that's not really well known for tardigrades. I mean, that's a beautiful question. I think, you know, you can kind of imagine like these dispersal events where you're moving between kind of really different environments, like say a fully aquatic environment to a partially aquatic environment. But then from that partially aquatic environment, as you disperse among different canyons and encounter different habitats and you become isolated from other populations, you might just be evolving due to that kind of those random processes that occur as you're isolated from other populations, or adaptation to local microhabitats. So it's really hard to say. That isn't really well known.
Okay. Question from Jennifer asking, what is the fossil record like for them?
So we have a few well-preserved fossils. So in general, you know, invertebrates, unless they have some hard parts to preserve, are poorly preserved. Now, luckily for tardigrades, they have their cuticle and they have their buccal apparatus, their mouth apparatus. And so we have a handful of fossils. Some are preserved in amber. Those are our best fossils. And they come from about 550 million years ago. So our fossils, you know, and they're variously preserved, but we know that it existed at least 550 to 530 million years ago.
Well, I know you saw Beth was on here and she's asking, do you have any students currently doing tardigrade research?
So Beth, yeah, you know, since I became the assistant vice president, I just have one student that worked with me recently, and he's this wonderful student who is a high schooler this year. And so I've been working with him off and on, but I'm hoping that I can recruit a couple students now that I've kind of gotten used to my AVP position. So.
Okay. And then a couple questions from Val. Why is the one species your favorite? I believe that was Testudo.
So McInnis is just so pretty under the microscope. It's bright red, and it's red because of the food that it eats. So it's feeding on, you know, it's feeding on plant matter, but the carotenoids in the plant matter are actually kind of preferentially sort of showing up. You can see right through the tardigrade, so I'm really in love with the color, but I also just love those hair-like structures. It kind of looks hairy to me. Just the morphology is very attractive to me. I love how they amble around. I love their little claws. They're very fascinating to me.
I wish I could see one.
Come on over.
I will.
Okay.
And then her second question was, are water bears found in all water across the planet?
So most of the water that we've sampled has them. They're not as common in fast running water. They really need some substrate, something to hold on to. And so still waters are better. They're kind of interesting because they can be even found in like hot springs. So another extreme environment that they can be found in. Some of them, you know, one of the most unusual places I found a tardigrade is one of my students and I traveled to northern Iceland together to look for tardigrades. And we looked at the tide pools in the northern edge of Iceland. And for days and days, we collected algae and we collected gravel and we looked and we didn't find anything. And finally, I started collecting barnacles. And right within the folds, the outer folds of barnacles, tucked in there were these tardigrades. They were absolutely amazing. So they can live in kind of sort of commensal with other animals.
Wow. And then a question that kind of connects to that one, are they found in Antarctica?
Yes, they are. So the lab that I got my training in, so I should tell you I'm actually a marine biologist who works on sea worms. and really enjoy sea worms, another obscure group of organisms. When I moved here, a few years after moving here, I realized I could study tardigrades as well, something local. And to really learn about them, I took a sabbatical and I worked with the lab that produces the greatest number of tardigrade papers in the world. And they actually had a study in Antarctica, and they discovered quite a few species in Antarctica. So they're all over the world. And interestingly, they're even found in glaciers. Some glaciers around the world, they form these little balls made up of moss and glaciers, and tardigrades live in them. So.
Wow, very cool. We have another question from Val, and she's asking, do we know how tardigrades interact with humans?
They don't really have much of an interaction with humans. They're not found on human bodies. They really stick to the environment other than humans. I don't think that question is really well known. They are affected heavily by humans. So when we go out and, damage crust, we are killing tardigrades. Radiation is very harmful to them. One of the most fascinating studies that I saw at the tardigrade conference that I attended was the effect of an irradiated body on a tardigrade. So not direct radiation, but let's say you had some sort of tissue that was exposed to radiation, and then you put that tissue or whatever in the habitat of the tardigrade. That tardigrade is deeply affected by that. So radiation that humans produce through energy or other forms can affect tardigrades. So.
All right, well, we have one more question, then we'll close things off. The last one's from Derek. Do tardigrades have an ecological purpose, decomposer, etc.?
Yes, so they basically play roles that you would imagine organisms in a forest play. So in a forest, we find herbivores. Some tardigrades are herbivores. We find carnivores. Some tardigrades are carnivores. We find scavengers. Some tardigrades are, now we know, are scavengers. So, the variety and complexity of the roles that we find animals play in larger ecosystems are all played out in smaller ecosystems, and we see tardigrades play some of those roles.
Very cool. Well, thank you everyone for the great questions. And thank you, Dr. Palmer, for the fascinating presentation. I'm excited to learn more about them in the future. So we do have one additional life science lecture series coming up on October 20th. Our last one is with Dr. Nikki Grant Hoffman, and she is the BLM ecologist for the National Conservation Areas, and she'll be talking about her fire restoration treatments that she has ongoing in McInnis Canyons NCA. So we're excited about that one, and you can find more information about that on our website. I just posted it in the chat box if you want to go to our website and kind of click around, see what we have to offer. Yeah, on our website you can find more about our programs. You can find additional community events that we're offering this fall, and you can find out ways that you can support our organization. So please reach out if you have any questions, and we hope to see you in the future. Once again, Dr. Palmer, thank you so much, and I hope everyone has a great evening.
Thank you. Good night.
Bye.